crew. This is Mr. Taliga, and we're doing a screencast for Cell Unit Life Science 7. This screencast serves a few purposes. First of all, this is happening towards the very end of this unit, and you should be working on your cell model if you haven't already been working on that. That's due here at the end of this week. This package should be just about done. And so if you go through the screencast with me and confirm and verify your answers, I think that's a good idea. If you need to complete some things, well, that's okay too. The purpose is to try and get this unit done. So the first few pages are, the table of contents basically for the unit. So there's nothing you need to check off things when you're done. Keep your packet in order. The next page 2 is the unit organizer. There isn't anything for us to do there. The third page is the cell model, and that too is something that is for you guys to work on, the plant versus animal cell. Remember, this is like a Venn diagram where the plant cell is the square one on the side, and then the animal cell is the kind of roundy one, and then the stuff in the middle is things that they both have. You'll need to fill that out as well. It's on Schoology, so you can look at that. I also have some pictures of plant and animal cells posted inside of the transitions class, actually near this screencast too. You can take a look at those, and if you haven't got this one filled out, you can use that to fill it out. So we're going to start actually on the 4th page of this packet. It says Cells in Life, and it's lesson 1. The page number on the bottom is actually page 15, and we're going to talk about some cell theory to start. So it says list 3 parts of cell theory. So inside of cell theory, the cell theory is all living things are made of one or more cells. So of one or more cells, and the cell is the smallest unit of life, so that's A. Whoops, let's worry about that. Cell is the smallest unit of life, and then the other part of cell theory is that all new cells come from pre-existing cells. Pre-existing cells move to cell division, which moves to new cells. So whoops. And existing cells cell division, dividing, and then on to new cells. Like that. Okay, so macromolecules and then descriptions. So identify and describe 4 types of macromolecules in cells by completing the table below. So nucleic acid are macromolecules that form long chains of molecules called nucleotides. nucleotides. I think I probably need to shrink that down and make it more like 18. And then I probably have to fix the spelling up on that. Yeah, okay, so there, I think that's it. I'll get this other one out of the way. Move that over a layer like that. Okay, that looks pretty good. Okay. I think I'm also going to move to full screen mode here too, just to make it easier for you to read. All right, and then proteins, the next one. And those are long chains of amino acid molecules. Lipids. Whoa, come on. Lipids are large macromolecules that do not dissolve in water. Oh, maybe I can't do that. Hold on. Okay, and then D is carbohydrate. And that's long chains of sugar molecules. So have your learning partner guess the main ingredient of all cells. And I bet you've already guessed it. The main ingredient of all cells is water. Okay. All right, and then on to the next page. That should be page 5 in your packet. I think its number is 17. The first thing it wants us to do is to check all scientists understand cell development. So how did scientists understand cell development? Put a check mark on the line before each item that helps scientists to develop cell theory. And building the first telescope is not what helped them, but definitely building the first microscope helped. Making more advanced microscopes, that also helped in developing cell theory. And then the invention of the electron microscope did not help with cell theory. It helped with other things, but not cell theory. 5, comparing plant and animal cells definitely helped. And then discovering protists did not help. So it would be 2, 3, and 5 that need to be check marked there. One-cell organisms. Is not a living thing. Single cell, we're choosing, so we're explaining why each of these statements is false in the cell theory. So the first statement is a one-cell organism is not a living thing. And that's false because a single celled organism is a living thing. And the cell theory states that living things are made of one or more cells. Maybe that one I can leave big. I probably can. Let's see here. Yeah. Okay. Let me get this out of the way. And then move that there. Okay. So the thing is that the cell theory states that living things are made of one. The keyword here is one or more cells. So we definitely, a one-celled organism can be a living thing. And here it said not. Right here, okay. Okay, so the smallest unit of life is the atom. And that's false. According to cell theory, a cell is the smallest unit of life. So okay. There you go. I'm just cleaning this up a little bit. Viruses are not cells, but they are alive. And this is also false because viruses are not cells, okay? So viruses are not living things because they are not cells. All right. Okay, and then plants do not come from cells, they come from seeds, and basically seeds contain cells, so yeah, that all does. The seeds come from cells, so that one is also false. So all cells, including plant cells, come from other cells, seeds, contain cells. Okay, on to the next page. So that was page 17. Now we're moving on down. This is content practice A, the cell. It is page 30. And the cell membrane. So the cell membrane protects the inside of the cell from the environment. And this one is just protects the inside of the cell from the environment blank the cell and it's just outside the cell. The cell wall protects cells from attack by harmful organisms. Cell appendages are often used for movement. Cytoplasm is a fluid inside the cell. And then the cytoskeleton forms a blank inside the cell. That's a framework. The nucleus is part of the eukaryotic cell that directs all the cell's activities and contains genetic material. All right, and then important molecules made of ribosomes are proteins. Okay. Endoplasmic reticulum. This is the production of proteins. Ribosomes attached a site. So an endoplasmic reticulum has ribosomes. Attached is a site of production proteins. And then mitochondria are the sites of energy producing. Processing. And then chloroplasts produce light energy. Carbon dioxide to make glucose. Sugar. and release oxygen. So the Golgi's apparatus prepares proteins for specific jobs. Vessels. Transport substances. And then vacuoles store waste. Vacuoles store food, water, and waste materials. Waste material. All right. So that's that one. Okay, that's page 30. Now on to the next page. This is the skill builder, the cell, and it is page 35. So we start with defining features of prokaryotic cell is that the genetic material is surrounded by a membrane. Other characteristics of prokaryotic cell is that they do not have all the cell parts. Found in eukaryotic cells. Most prokaryotic cells are one cell or unicellular organisms. Organisms are called prokaryotes. Another word for prokaryotes is bacteria. Eukaryocells make up plant, animal, fungi, and protists. These organisms are called eukaryotes. Almost all eukaryotic cells have genetic information. The cells is another membrane-surrounded component. Called organelles, which have specialized functions. It just has organelle specialized functions. Other differences between the prokaryotic and the eukaryotic cells are their size. Okay, and page 2. Go on to the cell. This is just a weird shaped Venn diagram. So basically, the top two boxes are the prokaryotic and the eukaryotic cells. And then the bottom is like kind of what they have in common. So maybe I can do a quick drawing of what I mean here. Let's see. Might have to zoom out a little bit and use a pen here. Grab a pen. Yeah. So it's kind of like this. This is what Miss Bout showed me today. If you think about how a Venn diagram has a shared bit in the middle, that's kind of like that bottom piece. So it's like this. You can do this if you want. You don't have to, but it's just a way to look at it. So like the side boxes are the things that are unique to the cells, and this bottom is the things that are that are shared. So this is the shared piece. These are the things that are just prokaryotic, and this eukaryotic cells are these things over here. That's a really messy thing, but all right. So that's what that is. Go back to full screen mode here. Okay. Oh, I'm going to need my tool different here. Probably would have given it to me if I'd done that. View full screen. Okay. So here are the things that the prokaryotic cell have that are unique or specific to them. It has genetic material. It has genetic material membrane. And then it also has does not have many cell parts. And then our bacteria. The order doesn't matter here. So, like, you know, if you got them in a different order, it doesn't matter. Okay, now the things that are specific to the eukaryotic cells. Have genetic material surrounded by a membrane. And then contains membrane surrounded organelles. And then makes up plants, animals, fungi, and protists. Okay. Then the stuff that they both have in common, they have cell membranes. Some have cell walls. They have cytoplasm. And they have a cytoskeleton. Whoa, can't find my cursor, there it is. Okay. Then that's page 36. And on to page 37 here. Or not 37, I'm sorry. This is the lesson outline moving cellular material. It's page 48. So this is a passive transport. Cell membrane. Is semi-permeable, which means that it allows only certain substances to enter or leave a cell. Passive transport is the movement of substance through a cell wall without using the cell's energy. So if it doesn't use any energy, it's passive. Diffusion is the movement of a substance from an area of higher concentration to an area of lower concentration. Usually diffusion continues through a membrane until the concentration of the substance is the same on both membranes. So this would be concentration. And then osmosis is the diffusion of water. Osmosis is the diffusion of water molecules only through a membrane if the concentration of water in the air surrounding a plant is less than a concentration of the water inside the plant's vacuoles. Water will diffuse into the air until the concentration of water is equal. Facilitated diffusion allows molecules to pass through a cell membrane using transport proteins. And carrier proteins carry molecules through a cell membrane. Channel proteins allow ions to pass through the cell membrane. Active transport uses cells energy to move substances through a cell membrane. Active transport moves substances from areas of lower concentration to areas of higher concentration. Okay, and then on to the next page. Oh, this is just the continuation of this. So a cell uses, is it endotosis and endoketosis? take substances by surrounding it with the cell membrane. So here's a picture of this coming up here, I think on the next page. A cell's vessel releases their content outside of the cell. This is exotosis, like exit, right? Exotosis, outside of the cell. Cell size and transportation. For a cell to survive, its surface area must be large compared to its volume. As the cell grows, its volume increases. Okay, next page. Here, moving cellular material. So if we're going to move some cellular material, we got to list the facts or terms next to each of the bullets to complete the chart. So, what passes through a cell using passive transport? Well, oxygen, carbon dioxide, other small molecules. Okay, what are 3 types of passive transport? Diffusion, osmosis, and facilitated diffusion. Oops. How do substances move in passive transport? From higher to lower concentration. And then what is the diffusion of water molecules only? It is osmosis. And then what is used to facilitate diffusion to assist the transportation of sugar and sodium molecules? Carrier proteins, channel proteins. Okay. And then what is only used in active transportation? And this is energy. Okay, and then how do substances move in active transportation? From lower to higher concentration. 8. Which proteins are used in active and passive transportation? Carrier proteins. And then what does a cell do to a substance? Nucleotides. Surround it and brings it inside the cell. And then 10, what does the cell use to eliminate? Exit. Exocytosis. And then which structures join the cell membrane and exocytosis? And that is vessels. All right. Next page. Skill builder. This is moving cellular materials. So there's descriptions and pictures here. We have to write short description of each transporting system. Under each description, we add a simple diagram that shows how the process occurs in a cell, including arrows. I'll do the best I can here. I'm not exactly an artist. You guys know that. I will try my best. Yeah. So the first one, the description is of diffusion, and that is the movement of a substance from an area of higher concentration to an area of lower concentration until equilibrium is reached. Alright. I'll leave it big and then I'll just slide it out so that's easier to get down. Yeah. So then, if we draw that one out, it kind of looks like this. So we have kind of two cells here, or two bodies. So we got like this one here, and then we got kind of like this one here. And the idea is, if we have a whole bunch of, like a tight concentration of items inside of this cell, and then not so much on the other side, that the material is going to move this way from high to low. Okay. All right. I'm going to make this one 16 to start with, and we'll see where we get. Osmosis is the diffusion of only water molecules until the equilibrium is reached. So I'll make a kind of the same diagram, but I'll talk about just water molecules. Actually, I might just do this. I'll just do like one cell and then going out. So like this, and then we got H2O, 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 and then we got them going like this. So it's just the transportation of H2O, and then they got some H2Os that are going the other way until balance is reached as far as the concentration goes. Okay. Then on this one, facilitated diffusion. Special transport proteins help molecules through a cell membrane. And basically what I did here is I made a cell, and then like here's my little transport proteins. They're like little cars. There they are. And these help to move things through. So they're molecules. So these are our proteins here, transport proteins. TPs for short. On top of them are molecules, MOLs. And so what's helping the MOL to move? It's the transport proteins, little cars, okay? So they're going this way. They're heading out that way. Woo, like that, okay? Now active transport. Cell membrane surrounds a material and takes it into the cell. And then the diagram, if you had yourself like an object, and then here the cell would kind of be like surrounding it, and it would kind of form like a mouth around it, and then it would just like be taking it into it, material surrounding. And then the description of the other one here is cell vessels, a cell's vessels joins with a cell membrane to release substance outside the cell. So that diagram kind of looks like this. So we have a substance inside of the cell that is joining with the substance outside of the cell, and then it's using that little creation as a gap for movement. Okay, and then the next page. Cells and energy. What does cellular respiration convert? Oops, hold on a second. Energy into food ATP. And then 2. Do that in every single one. That's going to be great. In which organelle does respiration take place? Mitochondria. And then sugar. Oh, what happens during glycosis? Broken down to smaller molecules. Some ATP produced. Let's move that one up a little bit. What 3 things are produced from respiration? Large amounts of ATP, water, and carbon dioxide. How does the process of fermentation differ from the process of respiration? Uses less ATP, occurs only in the cell's mitochondria. All right. I think I'm going to save, and I'll come back to this page. I'm just worried that my disk space is going to overload here. So I'll come back to this page and rejoin you in different